Hey there, how was your day? Wherever you are in the world, whatever time it is, I'm glad you're here. Maybe you're lying in bed or sitting somewhere quiet, trying to slow things down. Let's do that together. Tonight on HypnoScience, we're entering a world so small. You've never truly seen it, but it's been with you all along. It lives in your skin, in the folds of your breath, in the damp, quiet places the sun never touches. It's a world of invisible life, not alien, but strangely familiar. We're going to explore the inner workings of microbes, the soft clockwork of single cells, the way they eat, the way they breathe, the quiet mechanics of how they stay alive, in places too harsh for anything else. Some of them thrive in boiling acid. Others rest in glaciers, asleep for centuries. Many live in your stomach, breaking down breakfast with a patience you'll never notice. But they all share one thing: a rhythm, a deep, slow, biological rhythm. No brain, no heart, just chemistry humming in loops. This is the physiology of microbiology, and it's going to be very, very slow, which is exactly what we need. So if your eyes begin to close, that's all right. Let the hum of electrons and enzymes lull you. Let the world fade to something smaller and softer. And before we begin, if you feel like sharing, drop a comment with your time and location. It's oddly comforting to know when and where we're all tuning in from, or gently drifting off. This is HypnoScience. Let's go microscopic. Microbes are small, very small, so small that a single grain of sand could hide millions of them in its shadow. They slip between spaces in the world you never thought had room. A bacterium is usually 1 to 5 micrometers in length. That means you'd need 1,000 of them, lined up end to end, just to stretch across the head of a pin. Viruses are even smaller. They float like dust motes, invisible even under a regular microscope. But their size isn't a weakness. It's part of what makes them work. The smaller a cell is, the faster nutrients can reach every part of it. A short distance from membrane to center means a short trip for food, for waste, for signals whispering across the cell's interior. Their tiny size means they don't need complex systems. No blood, no bones, no lungs or liver, just a membrane, a few molecules, and a quiet kind of efficiency that larger organisms can't match. They live by the physics of surfaces. Surface area matters more than volume. Every micron of outer membrane is a gateway for sugars, gases, ions, water. The smaller the organism, the more surface it has compared to its inside. Imagine a balloon. If it gets larger, its volume grows faster than its surface. But for a microbe, staying small keeps everything close to the edge, close to the world outside. This means they respond fast to heat, to food, to toxins drifting by in a drop of water. Some change shape, some divide, some even die, quickly and quietly. Their simplicity hides sophistication. It's easy to assume that small means dumb or lifeless, but these cells are ancient, older than trees, older than bones, older than multicellular life. And in their simplicity is a kind of precision. Each structure in a microbe has a role. The outer membrane, thin but protective. The cytoplasm, a thick jelly-like sea, where reactions unfold in silence. There's no wasted space, no room for extras. Every piece of a microbial body must work, must function, must fit. Their size also helps them multiply. Fast, a bacterium can divide in as little as 20 minutes. Under the right conditions, that means one cell becomes 2. 2 become 4. 4 become 8. And so on. Quietly, exponentially, in just a few hours, one invisible speck can become a colony, a film, a bloom, sometimes helpful, sometimes harmful, but always alive. This rhythm of growth is part of their design. Small cells use little energy. They reproduce with little effort. And they can adapt, because with so many new cells, so quickly, mutations are common. And mutation is the first step to change. That's how they evolve. Fast and slow, all at once. In the stillness of a puddle, on the surface of your skin, inside your mouth while you sleep. They are testing variations. With every new division, each one is a quiet experiment, a silent trial. Most fail, some don't, and those that succeed, they keep going with the same calm, same shape, same size, nothing flashy, just persistence, through cold, through time, through everything. All of it begins with being small. All microbes have a boundary, a skin, a quiet edge between the inside and everything else. This edge is called the cell membrane. It's soft, thin, just a few molecules thick, but it holds everything in and keeps everything else out. Made mostly of lipids, fatty molecules that don't mix well with water. This membrane forms a double layer, 2 sheets facing away from each other, their tails tucked inside, their heads turned out. It's called the phospholipid bilayer. It moves like oil on water. Soft, flexible, always shifting but never breaking. In this soft wall, proteins float like islands. Some let nutrients pass through. Some act like doors, opening for one molecule, but not for another. Others act as sensors, detecting changes in temperature, pH, or nearby chemicals. The membrane is selective. It doesn't let just anything in. It chooses, gently, constantly, outside of this membrane. Many microbes have something more. A cell wall, thicker, sturdier, built to hold shape and withstand pressure. In bacteria, this wall is made of peptidoglycan, a mesh of sugars and amino acids. It's like a net, strong but porous. It keeps the cell from bursting. When water flows in, it holds the shape. Rod, sphere, spiral, whatever the microbe needs, to stay steady in its environment. Some bacteria have 2 membranes, one inside and one outside. Between them sits a thin wall, a sandwich of structure. These are called gram-negative bacteria. Others with just one membrane and a thick wall are called gram-positive, a simple name for a structural difference. Fungi, too, have walls, but theirs are built from chitin, the same tough material found in the shells of insects. Algae use cellulose, the same material found in plants. Even among microbes, there are small echoes of the larger world. Some microbes go without a wall. They are soft-bodied, fragile, flexible. They must live in safe places, inside hosts, or in environments that don't change too quickly. And then there are the strange ones, the archaea, ancient, unusual. Their walls are made of different stuff entirely. Not peptidoglycan, but pseudopeptidoglycan, or other molecules. They're adapted to places that feel like the beginning of the world. Hot springs, salt lakes, acidic pools, and deep ocean vents. But all of these variations, the walls, the membranes, the hidden architecture, have the same purpose: to separate life from not life, to hold in the slow chemistry that makes a cell alive, to regulate what comes in and what goes out, to create a quiet pocket of order in a chaotic, shifting world. These barriers are not static. They move, they repair, they stretch when needed, and shrink when food is scarce. The edge of a microbe is more than a shell. It's a decision-making surface, a place of choices, a line between inner and outer, between the small self and the endless everything else. Inside the membrane, beyond the soft boundaries and silent walls, is the world within, the inside of a microbe, a place without light, without sound, without air. This space is filled with a thick, watery substance called the cytoplasm. It is not empty. It is not still. It is dense, like slow-moving jelly. It holds everything the microbe needs to live. Not organs, but molecules, enzymes, proteins, ions, and small pieces of memory. All drift in this quiet soup. There is no separation here. No rooms. No compartments. Just soft organization, held together by chemistry and time. In the center, or sometimes off to one side, is the DNA. Not packed into a nucleus, not wrapped in velvet coils. Just a loop, a quiet floating thread of genetic code. It is called the nucleoid. It holds the instructions for everything the cell will do. How to make a protein, when to divide, what shape to grow, how to repair a wall. The DNA does not speak, but enzymes read it, slowly, methodically, turning chemical letters into action, one gene at a time. Nearby float the ribosomes, tiny factories made of protein and RNA. They build proteins, one link at a time, like threading beads onto a string. Each bead placed with purpose, each chain folded into shape. These proteins float free, or nestle into the membrane, where they become channels, or sensors, or silent workers, handling sugar and salt. Also scattered throughout the cytoplasm are reserves, granules of carbon, tiny bubbles of gas, crystals of sulfur, stored energy, stored possibility. Everything drifts, but nothing is random. Diffusion carries molecules from place to place. The cell doesn't need roads. It uses the natural flow. Enzymes meet substrates. Ions find channels. Signals move in invisible waves. Not loud, but certain. There is no heart, no lungs, but there is breath, in the form of respiration. A slow transfer of electrons from one molecule to another. Energy extracted, quietly, patiently, one reaction at a time. Every motion, every shift, every growth begins in this interior, this dim, formless chamber, where life happens, without noise. Sometimes a microbe will stretch. Its wall will soften at one point. The inside will grow. New membrane will form. The loop of DNA will copy itself, and one cell will become two. All of this begins here, inside, in the jelly, in the flow, in the rhythm of silent molecules, meeting and separating, binding and breaking. This is the hidden engine of microbial life. Not loud, not bright, but constant. Every moment, the inside hums. All living things need food, even the smallest, even those without mouths. Microbes do not eat like animals. They do not chew. They do not swallow. Instead, they absorb. Directly, silently, one molecule at a time. The outer membrane is not just a shield. It is a gate, a filter, a soft, intelligent surface, where choices are made. Some nutrients drift through on their own. This is called passive diffusion. Small molecules, like oxygen, slip between the lipids. Without resistance, they follow concentration, from where there is more to where there is less. No energy needed, just the quiet pull of balance. But not everything moves so freely. Larger molecules, like sugars or amino acids, need help. Here, the cell uses facilitated diffusion. Proteins embedded in the membrane act as doors. They open for the right shape, close for everything else. Still, other molecules move against the flow, from less to more. This takes energy. The cell must pay. This is active transport. It uses special proteins that burn chemical fuel, usually ATP, a small currency of energy, spent molecule by molecule. Some microbes bring in nutrients by folding in part of their membrane. They form a bubble around the substance, pull it inside, then seal the bubble. This is endocytosis. It is rare in bacteria, but found in some other microbes. Like amoebas, others release enzymes into the world around them. These enzymes break large molecules into smaller ones. The fragments drift back through the membrane. This is external digestion. Slow, chemical, without sound. Once inside, nutrients enter the cytoplasm. There they are sorted. Some are used for energy. Some become building blocks. Some are stored for later. Nothing is wasted. Microbes are efficient. They have to be. There is no room for excess. No space for delay. The environment matters. In nutrient-rich places, they grow fast. They divide often. In poor conditions, they slow down, or stop, or wait. Some microbes prefer sugar. Others prefer sulfur. Some eat iron, or nitrogen, or hydrogen gas. Each type is tuned to its own meal, a diet shaped by evolution, by place, by possibility. There are autotrophs, which make their own food from light or chemicals, and heterotrophs, which absorb the products of others. Some live alone. Some live in crowds, in mats, in films, in communities, where one cell's waste becomes another's feast. They eat in silence. They eat in rhythm. They eat without eyes, without hands, without hunger as we know it. But still they eat, not because they want to, but because they must. It is the slow pulse of survival, a motion too small to see, too soft to hear, but always present, always ongoing. Metabolism is life's quiet engine. For microbes, it is everything. A slow, steady flow of chemical reactions. At its heart, metabolism is about energy. How to capture it, how to store it, how to use it. Microbes have two main metabolic processes: catabolism and anabolism. Catabolism breaks down molecules. It releases energy. Anabolism uses energy. It builds new molecules. These two sides work together, like a slow, balanced dance. The energy currency of the cell is a molecule called ATP, adenosine triphosphate. Imagine it as a tiny battery, charged and ready. When the cell needs power, it breaks ATP apart. This releases energy. The cell uses this energy to do everything: repair walls, make proteins, move molecules, divide, and more. ATP is made through several pathways. One of the simplest is glycolysis, breaking glucose into smaller pieces. This happens in the cytoplasm. Glycolysis produces a small amount of ATP and other molecules that can be used later. If oxygen is present, microbes often continue with aerobic respiration. This is a longer process, taking place in membranes. It uses oxygen to extract much more energy. Electrons flow through a chain of proteins, the electron transport chain, passing from one molecule to another, like a slow-moving river. This flow pumps protons across membranes, creating a gradient. The cell uses this gradient to make large amounts of ATP. If oxygen is not available, some microbes switch to anaerobic respiration. They use other molecules, nitrate, sulfate, or even carbon dioxide, as the final electron acceptor. This process yields less energy, but it keeps the cell alive. In harsh conditions, there is also fermentation, a pathway that breaks down sugars without using an electron transport chain. Fermentation produces less energy, but it allows microbes to survive. When oxygen and other acceptors are missing, these processes happen slowly, quietly, without noise. Each reaction is a small step, part of a great chain. Energy moves through the cell, flowing, shifting, like a hidden current beneath still water. Metabolism controls the rhythm of life. It sets the pace for growth, division, and adaptation. Without metabolism, microbes would be still, silent, dead. But with metabolism, they hum, they pulse, they live. Glycolysis is the first step in breaking down sugar. It happens in the cytoplasm, the jelly-like interior of the cell. Glucose, a simple sugar, enters the cell quietly. No fanfare, no rush. The molecule is slowly split, step by step, into smaller pieces. Each step is guided by enzymes, proteins that act as tiny machines, folded perfectly to make chemical reactions happen at just the right pace. These enzymes lower the energy barrier, allowing reactions to proceed without noise or force. As glucose breaks apart, a small amount of energy is released. This energy is captured in molecules called ATP and NADH. ATP, the energy currency, stores power for the cell to use. NADH carries electrons to later stages of metabolism. This process does not require oxygen. It can happen in dark, quiet places, in the depth of a soil particle, or the folds of your gut. When oxygen is scarce, some microbes use fermentation to keep producing energy. Fermentation is a chemical shortcut. It recycles NADH back into NAD+, allowing glycolysis to continue. The end products of fermentation vary. Some microbes produce lactic acid, slowly souring their surroundings. Others release ethanol, a kind of alcohol. These products are not waste. They are signals, sometimes poisons, sometimes fuel, for other microbes nearby. Fermentation is efficient than respiration. It yields less ATP per glucose molecule, but it is a vital backup system. It allows microbes to survive in places where oxygen cannot reach. This quiet chemical dance keeps the microbial world alive. Slowly, patiently, every split sugar molecule, every ATP made, is a tiny step in a long journey of survival and adaptation. No dramatic explosions, no loud events, just a steady, calm rhythm of life at its smallest scale. Respiration in microbes is a quiet chemical conversation, a slow exchange of electrons between molecules inside the cell. In aerobic respiration, oxygen is the final recipient. It accepts electrons at the end of a chain, deep within the membrane. This chain is called the electron transport chain. It is a series of proteins that pass electrons gently from one to the next. As electrons move, they pump protons across the membrane. This creates a gradient, a difference in charge and chemical concentration. This gradient is like a small battery. It stores energy. When protons flow back across, they power an enzyme called ATP synthase. ATP synthase is a tiny machine. It spins slowly, adding phosphate groups to add molecules, creating ATP. This process is called chemiosmosis. It is silent but powerful. It produces most of the cell's energy. Aerobic respiration is efficient. It yields much more ATP than fermentation, but oxygen is not always available. In those cases, microbes can use other molecules to accept electrons. This is anaerobic respiration. Instead of oxygen, they use nitrate, sulfate, or even carbon dioxide. These molecules are less efficient acceptors, so less ATP is produced. But this process allows life to persist in places where oxygen cannot reach, deep in soil, in sediment, or inside animals. Microbes find ways to breathe. Some use sulfur compounds. Others reduce metals. Each pathway is a quiet adaptation to its environment. Respiration controls the flow of energy in the cell. It regulates growth, movement, and response to stress. Without it, microbes would be still, silent, without the slow hum of respiration. Life would stop, but with it, they pulse and thrive in every corner of the earth. Some microbes have a quiet way of making energy that depends on light. These are the photosynthetic microbes. And like plants, they don't have leaves or roots. No flowers, no stems. Just simple cells with special molecules that capture sunlight. At the heart of this process is a pigment called chlorophyll. It absorbs light gently, like a soft green filter. When light hits chlorophyll, it excites electrons. These electrons move slowly, starting a chain of reactions that ultimately produces energy. This energy is used to fix carbon. Carbon dioxide from the air is transformed into sugars. Sugars that feed the cell. Sugars that build structures. This process is called photosynthesis. Photosynthetic microbes include cyanobacteria, sometimes called blue-green algae, and certain types of algae themselves. Cyanobacteria were among the first organisms to use sunlight in this way. Billions of years ago, they began to fill the oceans and change the atmosphere. They produced oxygen as a byproduct, slowly, quietly, changing the world forever. This oxygen made it possible for more complex life to evolve. The light harvesting machinery inside these cells is delicate but efficient. It sits in membranes, folded into stacks, to capture as much light as possible. These membranes are called thylakoids. The energy captured here feeds the rest of the cell's metabolism. Photosynthetic microbes live in water, from oceans to ponds. They can also live on rocks, in soil, and even inside other organisms. Some form mats, thin, shimmering layers that ripple gently with the currents. These mats create microhabitats, tiny worlds where other microbes live, feeding off the sugars and oxygen produced. Photosynthesis is a calm rhythm, tied to the cycle of day and night. When light fades, the process slows. When dawn arrives, it begins again. This gentle pulsing of life has been happening for billions of years. A soft, persistent heartbeat beneath the surface of Earth's waters. Nitrogen is all around us, making up most of the air we breathe. But this gas is locked tight. It doesn't easily mix with water or most living things. Yet nitrogen is essential. It builds proteins, DNA, and the molecules of life. Microbes have a quiet secret. They can unlock nitrogen from the air. This process is called nitrogen fixation. Special bacteria, called diazotrophs, carry out this slow, delicate work. They use an enzyme called nitrogenase. This enzyme is fragile, sensitive to oxygen, and it works only under specific conditions. The enzyme carefully breaks the triple bond, holding nitrogen molecules together. This takes energy, a lot of it. Once broken, the nitrogen atoms combine with hydrogen to form ammonia, a form that plants and other organisms can use. This transformation is slow and precise. It requires protection. Often the bacteria live in root nodules on certain plants, like legumes. Inside these nodules, oxygen levels are low, creating the perfect environment for nitrogenes to work. This partnership between plants and microbes is ancient. It shaped ecosystems and allowed life to flourish. In places poor in nitrogen, nitrogen fixation is a quiet miracle. It turns invisible gas into the building blocks of life. Without it, life as we know it would not be possible. The process is slow, delicate, precise, just like microbes themselves. Movement is one of life's quietest whispers, especially in the realm of microbes. Though these tiny cells are invisible to the naked eye, they are far from still. They move through their microscopic worlds with gentle precision, propelled by delicate mechanisms evolved over billions of years. The ways microbes move are varied, but all are purposeful. Some glide smoothly across surfaces. Others spin and propel through liquids. Many navigate their environments by responding to chemical signals carried silently in water and air. The most familiar form of microbial movement is powered by flagella, slender whip-like appendages that spin like tiny propellers. These structures can be as thin as a hair. They can rotate hundreds to thousands of times per minute, propelling the microbe forward with subtle grace. A single flagellum or multiple flagella attached to one cell enable it to swim through fluid environments, pores in soil, drops of water, or the mucus lining of a host organism. The flagellum itself is a complex marvel, made primarily of a protein called flagellin. It's built as a hollow, helical filament, attached to a rotary motor embedded in the cell membrane, and powered by a flow of ions. This microscopic motor turns without noise, with a slow and steady rhythm that drives the cell onward. By changing the direction of rotation, the microbe can alternate between smooth, forward swimming and random tumbling, which helps it reorient itself. This simple mechanism is the foundation of microbial exploration. Beyond flagella, some microbes rely on pili, thin, hair-like appendages that do not rotate, but instead extend and retract to pull the cell forward. This twitching motility is slower and more deliberate than flagellar swimming. It is especially effective for moving across solid surfaces, rocks, host tissues, or biofilms. Pili function like tiny grappling hooks, anchoring to surfaces before pulling the cell forward. This movement is silent and subtle, but essential for colonization and infection in some species. Pili also play roles in attachment, communication, and DNA exchange, making them multifunctional tools for microbial life. Not all microbes rely on appendages to move. Some glide or slide along surfaces, using mechanisms still not fully understood. Gliding motility involve secretion of slimy substances, changes in cell shape, or surface proteins that interact with the environment. These movements are smooth and continue, resembling a leaf drifting slowly across a pond. Such motility often occurs in groups, where coordinated movement forms swarms or biofilms that ripple and shift as a living carpet across their substrate. This collective motion is an expression of microbial cooperation and resilience. Movement for microbes is never random. It is guided by chemical gradients in their surroundings, a behavior called chemotaxis. They sense the presence of nutrients, toxins, or signaling molecules, and adjust their swimming accordingly. When a beneficial chemical is detected, microbes swim toward higher concentrations. When harmful substances appear, they move away. The process is a slow, steady sampling of the world, a dance choreographed by receptors and signaling pathways, embedded in the cell membrane. Without eyes or brains, microbes make decisions chemically, tuning their motor activity to navigate complex and changing environments. Microbes face many challenges in moving through their microscopic worlds. Fluids vary in thickness. Some microbes live in watery films. Others inhabit thick mucus or viscous soils. These differences affect how they swim or glide. The physical forces of fluid dynamics operate differently at such small scales. What is easy for a fish to swim through is more like moving through honey for a microbe. Their motility, structures, and behaviors are adapted to these challenges, enabling them to persist and explore, even in difficult conditions. Motility is an energy-intensive activity. Rotating flagella, extending pili, producing slime, all require a continuous investment of cellular energy. Microbes balance the cost of movement with the necessity of finding food, escaping threats, or colonizing new niches. When nutrients are scarce, they conserve energy by reducing movement or entering dormant states. The investment pays off when movement leads to better environments, richer resources, or safer habitats. Many microbes do not move alone, but as coordinated groups. Swarms of bacteria can form waves and patterns on surfaces, communicating chemically to synchronize their motion. These groups create biofilms, complex, multi-layered communities of cells, embedded in self-produced matrices. In these living carpets, microbes share nutrients, exchange genetic material, and protect each other from harm. Movement in such communities is a collective behavior, a silent conversation expressed through subtle shifts and flows. Consider the elegance of these microscopic motions. A single flagellum's slow rotation, the retraction of a pilus, like a delicate grappling hook, a gentle glide powered by secreted slime. Each is a soft thread in the fabric of microbial life. Each turn, each pause, each direction chain is a response to the unseen world of chemicals and physical forces. No eyes watch this dance. No brain directs it. Only molecules and energy flows shape the movements. Yet within this simplicity lies profound adaptability, a constant push to survive, grow, and evolve. The microscopic world is alive with motion too small to see, but too vital to ignore. It is a place where persistence meets precision, where life is a continuous journey. through unseen spaces. These tiny movements shape ecosystems, influence human health, and sustain the cycles of nature. As you breathe quietly, imagine the millions of microbes around you, moving through their invisible realms, with calm determination, carrying the slow pulse of life forward. In silence, microbial life is not just about movement. It is also about communication, a quiet exchange of chemical messages, invisible to us, but essential for survival. Microbes live in communities, whether in soil, water, or inside animals. They are rarely alone. And when together, they need to coordinate. Without voices, without eyes, without brains, they communicate chemically. This is called quorum sensing. Quorum sensing is a way for microbes to detect their numbers, to know when they are many or few. They produce and release small molecules, called autoinducers, into their environment. As more microbes release these molecules, the concentration in the surrounding fluid rises. When the concentration reaches a threshold, it triggers a response in the cells. This can switch on genes, leading to changes in behavior. For example, microbes may produce toxins, form biofilms, or move collectively. Quorum sensing is a form of social behavior. It allows microbes to act as a group, rather than isolated individuals. This group action can be crucial. Biofilms, for example, are complex communities where microbes protect each other and share resources. Without communication, biofilms would not form. They are essential in natural ecosystems and also in human health. Some infections involve biofilms. These are harder to treat because the microbes work together. Microbial communication also involves other signals. Some use electrical signals, sending tiny waves through their communities. Others exchange genetic material, sharing traits that may help survival. Microbes use chemical gradients to move toward nutrients or away from danger. This sensing of the environment is continuous, slow, and subtle. Microbes respond to light, temperature, and pH, adjusting their physiology accordingly. The complexity of microbial communication challenges our ideas of intelligence. Though lacking neurons, microbes display coordinated behavior. They solve problems, adapt to changes, and survive harsh conditions. This silent conversation is ongoing, a never-ending flow of signals, passing between cells, shaping communities, and influencing the world at large. When you breathe, you inhale a cloud of microbes that interact, communicate, and live in this way. Their world is rich with unseen connections. Imagine these countless dialogues taking place in every drop of water, every speck of soil, inside every living creature. Microbial communication is the heartbeat of their social lives, a slow and steady pulse that binds single cells into communities. Reproduction is the quiet rhythm of life. For microbes, it is the slow, steady pulse that carries their existence forward. And like animals or plants, microbes reproduce without flowers or eggs. They multiply simply, usually by splitting into two. This process is called binary fission. Imagine a single cell, a tiny sphere or rod, growing quietly in a drop of water or on a speck of soil. Inside, its DNA, the molecule of life, has already made a copy of itself. This copy floats beside the original, like two threads of silent instruction, waiting patiently for the next step. The cell begins to elongate, slowly stretching like soft dough. A new membrane starts to form in the middle, pushing inward, creating a dividing wall. This wall thickens and strengthens until the cell is pinched into two separate cells. Each new cell contains a full copy of the DNA, ready to live, grow, and eventually divide again. This process is remarkably efficient. Under ideal conditions, some bacteria can divide every 20 minutes. That means one cell can become over 1 million in just a few hours. Silent and invisible, yet powerful in their numbers. However, the process is not always this fast. Environmental factors like temperature, nutrient availability, and toxins can slow or stop division altogether. Some microbes can enter dormant states, waiting patiently until conditions improve. Others produce spores, tough, resistant structures that protect their genetic material for years or decades. Binary fission is not the only way microbes reproduce. Some use budding, where a small new cell grows from the surface of the parent and then detaches. Others exchange genetic material through a process called conjugation. Though not reproduction in the traditional sense, it increases genetic diversity. This diversity allows populations to adapt to changing environments, resist antibiotics, and survive challenges. Reproduction in microbes is a quiet, constant process. It is life unfolding without fanfare, without ceremony, just the slow turning of molecular machines inside invisible cells. Each division is a new beginning, a chance for survival, a step in the endless dance of life. Microbes face many challenges in their tiny worlds. Harsh environments, lack of nutrients, and sudden changes in temperature or moisture threaten their survival. To endure these hardships, some microbes have developed remarkable strategies. One of the most resilient is the formation of spores. Spores are like tiny, tough seeds. But unlike plant seeds, they are not designed to grow immediately. Instead, spores are built to withstand extreme conditions and wait patiently until the world becomes hospitable again. When a microbe senses danger, it begins a slow transformation. Inside the cell, a copy of the DNA is wrapped tightly. Protective layers of protein and other molecules form around this precious cargo, creating a shell that resists heat, desiccation, radiation, and chemical assault. This spore is dormant. Metabolism slows almost to a stop. The cell's machinery rests. Time seems to pause. Spores can survive for years, even centuries, hidden in dust or soil, waiting for moisture or nutrients to signal that life can resume. When conditions improve, the spore slowly rehydrates. It breaks open. The cell inside awakens, and the microbe begins life anew. Not all microbes form spores. Some use other survival tactics. Many adjust their metabolism. They slow their growth, reduce energy consumption, enter states of dormancy without forming spores. Others build protective layers around their cells, thicker walls, extra membranes, or slime layers that shield them from harm. Microbes can also change their behavior. They form biofilms, sticky communities of cells embedded in a self-produced matrix. These biofilms protect members from antibiotics, predators, and harsh environmental conditions. Within these communities, microbes share resources and communicate, increasing their chances of survival. Some archaea live in boiling hot springs or acidic pools. Their cell structures and enzymes are specially adapted to tolerate extreme heat and acidity. Others thrive in frozen tundras, where water is locked as ice. They survive by producing antifreeze proteins and entering metabolic slowdowns. All of these strategies reveal the incredible resilience of microbial life. Despite their small size, microbes are masters of endurance. They persist through time, through change, and through adversity. Their survival is a testament to the power of simplicity and adaptability. Microbes live in a world defined by conditions. Temperature, pH, salt concentration, all shape where they can survive, how they grow, and what they become. Each microbe has its own comfort zone, a range of conditions where life feels just right. Outside this zone, survival becomes a challenge. Growth slows, metabolism shifts, and some microbes enter dormancy. Temperature is one of the most important factors. Some microbes prefer moderate warmth. Around the temperatures humans find comfortable. These are called mesophiles. Others thrive in cold environments. Psychrophiles live in icy waters, deep oceans, and polar regions. Their enzymes work slowly, designed to function in the chill. On the other end, thermophiles enjoy heat. Some live in hot springs, volcanic vents, and other extreme places. Their proteins are stable at temperatures that would denature others. Beyond temperature, pH influences microbial life. Some microbes thrive in neutral conditions, neither acidic nor basic. Others prefer acidic environments. Acidophiles live in environments like sulfur springs or acidic soils. Their cell membranes and enzymes are adapted to protect against harsh acidity. Alkaliphiles, on the other hand, prefer basic conditions. They can be found in soda lakes or alkaline soils. Salt concentration is another key factor. Halophiles flourish in salty environments, such as salt lakes or evaporating ponds. They regulate their internal chemistry to balance the osmotic pressure. Microbes constantly sense these external conditions. They adjust gene expression, produce stress proteins, and modify membrane composition. These adaptations allow survival, even when conditions fluctuate. In natural environments, conditions rarely remain constant. Microbes experience cycles of temperature changes, pH shifts, and varying nutrient availability. Their ability to sense and respond to these changes is vital for continued life. Some microbes produce heat shock proteins, which help refold damaged proteins when temperatures rise suddenly. Others synthesize compatible solutes, small molecules that protect cellular components during salt or drought stress. The interplay between microbes and their environment is a slow, dynamic dance. Each microbe exists in a niche, defined by these physical and chemical parameters. The diversity of microbial life is, in part, a story of adaptation to this variety of conditions. This slow adaptation process has allowed microbes to colonize nearly every habitat on Earth, from deep-sea vents to Arctic ice, from acidic mine drainage to alkaline deserts. Microbes persist. Their ability to thrive in diverse environments demonstrates the resilience and flexibility of life at its smallest scale. Microbes rarely live alone. More often, they gather together, forming communities. One of the most fascinating examples of microbial cooperation is the biofilm. A biofilm is a slimy, sticky layer where microbes live side by side, embedded in a self-produced matrix of polysaccharides, proteins, and DNA. This matrix holds the community together, protecting it from the outside world. It is a shield against antibiotics, predators, and harsh environmental changes. Biofilms can form on almost any surface: rocks in a stream, the inside of a pipe, teeth, or even medical devices implanted in the body. Within the biofilm, microbes communicate. They share nutrients. They exchange genetic material. They coordinate defense strategies. This living city operates slowly, with intricate chemical signaling, guiding the behavior of individual cells. The matrix itself is a complex structure, porous enough to allow nutrients and gases to flow through, yet dense enough to provide protection. Biofilms exhibit gradients of oxygen, nutrients, and waste products. Cells in the outer layers often differ from those deep inside. This spatial organization creates microenvironments, allowing diverse species to coexist. Formation of a biofilm begins when free-floating microbes attach to a surface. They produce sticky substances to anchor themselves firmly. Once attached, they begin to multiply. The matrix develops, and other microbes join the growing community. This process can take hours to days, gradually building complex, multi-layered structures. Biofilms have important roles in nature. They participate in nutrient cycling, helping break down organic matter. They stabilize soils and sediments. In human health, biofilms can be double-edged. They protect beneficial microbes in the gut, but can also harbor harmful bacteria that resist treatment. Understanding biofilms is a key area of research. Scientists study their formation, communication, and resistance mechanisms, seeking ways to manage them in medical and industrial settings. Despite their slimy appearance, biofilms represent a remarkable form of microbial life. They show how simple cells can come together, cooperate, and thrive as a community. This cooperation provides resilience, allowing microbes to survive in conditions that might be lethal alone. In this slow, quiet way, biofilms illustrate the power of unity. In the microbial world, enzymes are the silent artisans of every microbe. They build, they break, they rearrange. Without them, nothing happens. Inside a single cell, billions of molecules drift. Left alone, most would rarely meet. Even if they touched, they might not react. The energy needed would be too high. Time would stretch into geological ages. Enzymes change that. They are folded chains of amino acids. They form pockets, tunnels, grooves. These shapes cradle other molecules, substrates, like gentle hands. The fit is precise. One twist, one bump, one missing atom, and the reaction fails. When a substrate nestles inside, bonds flex. Electrons shift. The energy barrier falls. A path opens that was closed a heartbeat before. The reaction glides forward. Products form, drift away, and the pocket stands ready for the next visitor. Every pathway in a microbial cell is a trail of enzymes. Glycolysis relies on 10 of them, each waiting for its moment. Respiration uses dozens more, arranged like beads on an invisible thread. Each bead pulses in turn, passing electrons, pumping ions, forging ATP. In photosynthetic microbes, enzymes capture photons, split water, fix carbon. In nitrogen fixers, the fragile nitrogenase eases apart triple bonds older than mountains. Some enzymes float free in the cytoplasm. Others anchor in membranes, half outside, half inside. Still others pack together into massive complexes, spun like carousels or stacked like coins. Each location, each grouping shapes the flow of chemistry. Temperature touches enzymes first. Heat loosens their folds, cold stiffens them. A thermophilic enzyme flexes comfortably at boiling, while a psychrophilic one would unravel. Evolution tunes every curve and hinge to local climate, making life possible from vents to ice. pH is another sculptor. Excessive acid or base tugs at charged side chains, opening or closing pockets. Acidophilic microbes craft enzymes whose surfaces are armored against proton floods, while alkaliphils weave negative charges into their cores to keep structure tight. Salt pulls water away from proteins. Halophils stud their enzymes with acidic residues, attracting a thin shell of water, even in brine. Without this trick, the proteins would clump and cease. Metal ions act as cofactors. A tiny bit of iron or magnesium slips into an active site like a key. Without the metal, the lock will not turn. Some microbes hoard these ions with siderophores, molecular lures that bind iron tighter than rust. Others store metals in protein cages until the moment of need. Not all enzymes work continuously. Cells regulate them with exquisite care. An end product may dock at a side pocket, telling the enzyme to slow. Phosphate groups may be added or removed, like on-off switches. Small RNAs can silence the genes that encode the enzymes, stalling production before the first amino acid is joined. Under starvation, microbes deploy scavenger enzymes. These chew worn proteins into amino acids, recycling precious nitrogen and carbon. During oxidative stress, repair enzymes patrol DNA, snipping out damaged bases and patching the strands. Motility depends on enzymes too. Flagellar motors run on an enzyme that converts proton flow to mechanical spin. Pilus retraction hinges on at pace enzymes that grasp and pull the filament inward. Without these motors, the cell would drift aimlessly in the dark. Communication is enzymatic as well. Autoinducer synthases stitch small signaling molecules. Receptor enzymes break them down when the message must fade. The rise and fall of these signals guide biofilm formation, virulence, dispersal. Some enzymes carry memories. CRISPR-associated nucleases snip foreign DNA, then tuck a snippet into a genomic archive. Later, the same nucleases read those memories and cut invaders on site. A molecular immune system, silent and specific. In archaea that breathe sulfur, enzymes pass electrons to elemental sulfur, forming sulfide in the dark. In methane-eating bacteria, methane monooxygenase splits CH4, turning a greenhouse gas into food. Each of these feats happens at ordinary pressures and temperatures, conditions chemists struggle to mimic. Turn to decomposition. Cellulases from soil microbes unwind tough plant fibers. Ligninases open the aromatic knots of wood. Chitinases nibble at the shells of insects and fungi. The world would choke on dead matter without these hidden workers. Speed varies. Some enzymes finish a cycle in 1,000th of a second. Others, like nitrogenase, crawl through a few dozen reactions per minute, because the energy cost is vast. Yet even slow enzymes outperform uncatalyzed reactions by factors of millions. Precision matters. DNA polymerase inserts the right base most of the time, but mistakes slip through. Proofreading enzymes follow, excising mismatches. In the rare event both fail, repair enzymes later scan for bulges and breaks. Thus the genome survives generations, with only a whisper of error for evolution to use. Enzymes cooperate. Product of one becomes substrate of another. Metabolic channels sometimes form, a handoff from pocket to pocket without spilling intermediates. This reduces loss, speeds flux, regulates flow. Energy status feeds back. High ATP may inhibit glycolytic enzymes, signaling plenty. Low ATP relieves inhibition, urging the cell to burn glucose. AMP, ADP, NADH, acetyl-CoA. Each molecule is a word in the chemical language. Spoken by enzymes, heard by enzymes, obeyed by enzymes. Some microbes secrete enzymes outside. Exoenzymes digest polymers into absorbable pieces. Pathogens use hemolysins to rupture host cells, freeing iron. Fungi extend hyphal tips loaded with enzymes that soften wood ahead, allowing growth through stubborn trunks. Industrial biotechnologists borrow these talents. They harness the most stable DNA polymerases in PCR. They ferment beer with yeast enzymes. They break laundry stains with bacterial proteases. Yet the natural roles remain unchanged inside the microbes. Day and night, century after century. Consider mutation. A single amino acid change can widen an enzyme pocket. Suddenly, a new antibiotic no longer fits. The microbial cell survives and multiplies, spreading resistance through plasmids and phages. Horizontal gene transfer spreads enzymes too. A gene for beta-lactamase leaps from one species to another, arming both against penicillin. Conjugation, transduction, transformation, each is an enzymatic act at heart. In deep subsurface rocks, where nutrients are scarce, enzymes are sparse and slow. Cells there may divide once each century. Their enzymes drift in viscous cytoplasm, waiting for occasional pulses of hydrogen or sulfate. In hydrothermal vents, enzymes operate near 120 degrees C. Their backbones are rigid, tied by extra ionic bridges. One wobble, one breath of cool water, and activity falters. Yet in boiling brine, these proteins churn happily, generating energy that supports entire vent ecosystems. Viruses, although not alive by strict definitions, carry enzymes too. Reverse transcriptase copies RNA into DNA. Integrase stitches viral DNA into host chromosomes. Protease cleaves long polypeptides into mature capsid parts. Without these enzymes, viruses would be silent fragments of code. Enzymes leave fingerprints in geology. Isotopic ratios in ancient rocks reveal past enzymatic pathways. Nitrogen fractionation betrays nitrogenase. Light carbon hints at methanogenesis. Thus the whispers of enzymes echo across billions of years. Future medicine may harness engineered enzymes that seek out and destroy tumor cells, or digest plaques in arteries. Yet wild microbes already wield such power for their own ends. Cutting, mending, sculpting at the molecular level, At night, while you rest, enzymes in your gut microbes ferment dietary fibers, releasing short-chain fatty acids that soothe inflammation. In soil beneath your window, enzymes cycle nitrogen, feeding roots that anchor trees. In oceans unseen, enzymes of phytoplankton fix carbon, producing half the oxygen you breathe tomorrow. Each catalytic act is quiet. No sound, no light, only the soft rearrangement of atoms. One spark of activation energy is removed. A bond forms or breaks. Heat dissipates into the surrounding water. The enzyme waits, ready again. Billions of enzymes echo this cycle each second in one cell. Multiply by the number of cells in a milliliter of seawater, then by all the seas. And the mind grows still. Enzymes are poetry without words. They write the story of life in invisible strokes, line after line, reaction after reaction. Close your eyes. Imagine those strokes continuing, steady as heartbeat, soft as breathing, through every microbe on Earth. Sleep knowing that in darkness and in light, these tiny artisans keep watch, maintaining the quiet machine, Rye of existence, with infinite patience. Inside every microbial cell, a loop of DNA rests like a silent scroll, tucked into the cytoplasm, coiled but unbound by a nucleus. It waits, patient, timeless. The letters on that scroll never speak aloud. They rely on messengers. And the first messenger is transcription. An enzyme called RNA polymerase settles onto the DNA. It feels for a special landing pad, a promoter. Often the promoter is marked by a sequence like 35 and 10 boxes. Tiny signposts only enzymes can read. When the fit is right, RNA polymerase eases the double helix apart. One strand becomes a template. Free nucleotides drift in the cytoplasm like loose beads. They snap into place. A UGC, one by one, forming an RNA chain. This RNA is a working copy, disposable. It carries the code outward. The DNA closes behind, untouched, eternal. Transcription in microbes is fast. Dozens of nucleotides every second. Multiple polymerases can travel the same gene at once, like quiet freight trains on a single track. Yet the cell is cautious. It does not transcribe everything at once. Control is vital. Sigma factors guide RNA polymerase. Each sigma binds certain promoters. Under heat, the heat shock sigma awakens. Under starvation, an alternative sigma rises. In this way, the cell swaps lenses, seeing only the genes it needs. Beyond sigma factors lie repressors, proteins that perch on DNA, blocking the track. The classic repressor is lac-I. When lactose is scarce, lac-I binds the operator. RNA polymerase cannot pass. The genes for lactose digestion stay silent. When lactose appears, it slips into the repressor's pocket, nudging lac-I off the DNA. The path clears. Polymerase moves on. Enzymes for lactose breakdown are made. Supply answers demand with no wasted effort. Some genes wait for activators instead. RSE curls DNA into a loop when arabinose is missing. Add arabinose, RSE straightens. RNA polymerase can now begin. A hinge of sugar decides the fate of a pathway. Farther downstream, the fresh RNA finds ribosomes. Protein factories float freely or stud membranes. Each ribosome grasps the RNA like a ribbon. It reads sets of 3 letters, codons. Every codon calls a tRNA. Each tRNA carries one amino acid. The growing protein threads into the cytoplasm, folding as it emerges. Chaperones cradle the chain, preventing premature knots. Hydrophobic cores tuck inward. Charged edges stay outside. Within moments, a working enzyme is born. Translation, too, is policed. Sometimes a ribosome stalls. Quality control enzymes detect the pause. Cut the half-made peptide. Recycle the ribosome. And discard the faulty message. Regulation can happen even earlier. Some RNAs fold into riboswitches. When a small metabolite binds, the RNA changes shape. A terminator stem may form, halting transcription, or a ribosome binding site may hide, blocking translation. Thus, the script edits itself mid-speech. Small regulatory RNAs patrol the cell. They pair with target mRNAs, marking them for destruction or preventing ribosome binding. These whispers of RNA silence unwanted songs. Environmental cues reach the genome through two-component systems. A sensor kinase straddles the membrane, feeling heat, pH, salt, or chemicals outside. When triggered, it transfers a phosphate to a response regulator. Phosphorylated, the regulator binds DNA. Genes switch on or off within minutes. Quorum signals join the chorus. As cell density rises, autoinducers accumulate. Luxar-type regulators sense the buildup. They activate genes for light in Vibrio, for virulence in Pseudomonas, for competence in Streptococcus. Community voices rewrite individual scripts. Stress can carve fleeting marks on DNA. Methyltransferases add small methyl groups to cytosine or adenine. Methylation can hide promoters or invite regulatory proteins. Patterns may persist after division. A soft microbial epigenetics guiding future generations. During DNA damage, the SOS response flares. Recce senses single-stranded DNA. It lifts the Lexay repressor away from dozens of genes. Repair enzymes flood the cell. Error-prone polymerases appear, willing to copy through lesions at the cost of fidelity. When the crisis passes, Lexay rebuilds repression. Calm returns. Horizontal gene transfer writes entirely new chapters. A plasmid enters through conjugation. Phage DNA injects like a silent dart. If welcomed, foreign genes acquire promoters, ribosome sites, and regulators. They join the operatic ensemble, adding powers the host never knew. CRISPR arrays record these invasions. Short spacer sequences, memories of past phage, sit between repeats. Cas proteins transcribe them into guide RNAs. When a matching phage returns, Cas9 cuts the intruder's DNA. Defense by remembering. All these layers, sigma choice, repressor locks, activator keys. Riboswitch folds, small RNA veils, two-component relays. Quorum merm. ERS methyl marks, SOS flares. And CRISPR sentinels form a network dense as forest roots. Signals weave through roots, turning genes up, down, sideways. Yet the overall rhythm stays slow, steady, quiet. A cell might activate a nitrogenase operon only at dusk, when oxygen drops, or silence motility, genes inside a host to avoid detection. Even in dormancy, regulation persists. Spores pack DNA with small acid-soluble proteins. They bind and bend the helix, shielding it from heat and desiccation. When water seeps back, proteases digest the protectors. Transcription resumes. Life unpauses. Consider the elegance of feedback. ATP levels fall. AMP rises. An enzyme senses AMP. Opens a promoter. Genes for catabolism serve. Energy is restored. AMP declines. The promoter closes. No overseer required. Only molecules reading molecules. Every second, thousands of transcripts bloom and fade in one bacterium. Most last mere minutes before RNases shred them. Proteins linger longer, hours or days, but are eventually tagged and dismantled by proteases. Turnover keeps the cell responsive, light on its feet. Gene expression in microbes is like candlelight in a dark room. Gentle, flickering, yet able to fill the space with warmth. The genome supplies the wax. Regulators trim or lengthen the wick. Metabolism feeds the flame. If you could shrink and listen, you would not hear clamor. You would hear a hush pierced by soft clicks. Polymerase starting. Ribosome shifting. Repressor landing. Activator lifting. Those clicks compose a score older than mountains. A score that guides microbes through feast and famine. Heat and ice. Light and dark. Alone and together. And as you settle deeper into rest, know that in every corner of air and soil, this molecular music continues. Unseen, unending. Telling each microbe when to wake, when to build, when to pause, and when to dream in silence. Stress is a word made for larger creatures. Yet microbes feel its weight in every moment. Heat, cold, acid, drought, poison, hunger, light, darkness. Each arrives like a quiet knock on the membrane. The cell listens. It answers in chemistry, never in panic. A sudden rise in temperature loosens delicate folds. Proteins quiver. Hydrogen bonds stretch. Left unchecked, shapes collapse. To guard against this, microbes launch the heat shock response. Sigma 32 awakens. RNA polymerase changes companions. New transcripts bloom. HSP 60, HSP 70, CLPB, GROL, DNA-K. These chaperones cradle trembling proteins, refolding strands, nestling hydrophobic regions back inside, buying time until the fever passes. Proteases follow with gentle mercy, cutting beyond repair, feeding fragments into the amino acid pool, where life will weave them anew. Cold arrives more slowly. Membranes stiffen. Enzyme rates lag. The cell slips into viscosity like syrup on ice. Cold shock proteins bind RNA, keeping messages flexible. Desaturase enzymes slip double bonds into fatty tails, softening the bilayer, restoring the ripple that lets nutrients flow. Ribosomes shuffle composition, trading subunits that work at low heat. Nothing frantic, only quiet adjustment. Oxygen once turned the planet blue, but in excess, it is a spark for chaos. Radicals bloom from leaking flavins and quinones, scratching DNA, denting lipids, rusting iron. When oxidative stress rises, Soxar's iron, sulfur center feels the bite. It oxidizes, grips DNA, summons SOXS and a legion of detox genes. Superoxide dismutase to tame O. Catalase to split H2O2 into water and breath. Peroxidexins to polish the last traces of flame. Small thiol molecules, glutathione in some, bacillithiol in others, sweep through cytoplasm, donating electrons like calming words. Acid slips through walls in quiet protons. They threaten to drown cytoplasmic order. To resist, microbes expel H+ with ATP-driven pumps. They import K+ to balance charge. Acidophils line their membranes with tetraether lipids, forming a skin almost without pores, keeping the inner pH near neutrality while outside boils in acid. If protons breach, decarboxylase enzymes bite amino acids, burning them to CO2 and ammonia, soaking up acid like chalk on vinegar. Alkaline shock is gentler but still a shift. When O- creeps in, cells pull in protons with Na plus H plus antiporters. They lace proteins with acidic side chains, anchoring hydrogen where it is rare. Peptidoglycan may thicken, a sturdier wall against slippery bases. Salt is thirst in disguise. Outside ions rise, water flees the cell. To hold it fast, microbes gather compatible solutes. Glycine, betaine, proline, ectoin, trehalose. These small molecules crowd the cytoplasm without disturbing proteins. Transporters swing wide. Biosynthetic enzymes hum. Water returns, and the cell keeps its form. Starvation dims all forces. Carbon wanes first. ATP drops. AMP climbs. Rel A and SPO T-sense, rising uncharged tRNAs at stall. D ribosomes. They spike the cytoplasm with PP-GPP. The alarmin of want. RNA polymerase rewrites priorities. Growth genes down. Scavenging genes up. Ribosomes shed proteins, shrink weight. Membranes tighten. Division stops. The stringent response is not despair, but fasting, a pause until the next faint whiff of sugar. Phosphate may vanish. Fer touches its periplasmic feeler, finds scarcity, phosphorylates Fer B. Fer B opens operons for phosphatases, teasing PO4 from DNA, lipids, soil minerals. Sulfur scarcity lifts size B. Iron lack freeze for repression. Zinc need wakes Sir. Each ion, each element has its sentinel. DNA damage cues the SOS. A UV photon snaps a thymine dimer. Replication stalls. Single strands bloom. Rec-A binds the exposed helix. Triggers Lex-A to cut itself. Repression melts from 40 genes. Nucleases, polymerase V, ligases photolyze. Repair floods in. Errors creep, but better flawed than broken. When the strands are whole, Rec-A releases. Lex-A builds again. The cell exhales. Toxin, antitoxin pairs wait as final fuses. Mas-F, Rel-E, HOC, toxins in dozens of families. Sit sheathed by fragile antitoxins. Stress tilts the balance. Proteases nibble antitoxins first. Toxins cut mRNA, block ribosomes, pierce membranes. Metabolism slows to near stillness. If conditions brighten, new antitoxins rise. Sheath blades, and life resumes. If not, the cell drifts into programmed rest or self-sacrifice, freeing nutrients for kin. Some microbes weave multiple stresses into one mode. The general stress response. In gram-positives, sigma B sits bound to anti-sigma RSBW. Shock shakes a phosphatase cascade. RSBV steals RSBW. Sigma B escapes. Opens over 100 genes. For osmoprotection, oxidative shield, acid defense, protein repair. In gram-negatives, RPOS, sigma S, fills a similar role, rising as growth slows. Preparing stationary phase armor. Biofilms cloak stress further. Deep layers feel anoxia, starvation, waste acids. They answer with persister cells, slow, drug-tolerant forms that ignore antibiotics. Tiny pumps spit toxins out. Matrix polysaccharides limit diffusion. If outer cells die, inner cells persist, ready to bloom when danger fades. Archaea invents no extremes as routine. Heat-stable chaperonins form barrel chambers. DNA is wrapped by histone-like proteins. Tight and safe. Reverse gyrase adds positive supercles. Locking the helix against heat unwind. Cranarchaeal membranes, where tetri the lipids joined tail to tail, forming monolayers that shrug at boiling brine. In the cryosphere, psychrophiles fight ice. Antifreeze proteins bind tiny crystals, halting growth. Cold-active enzymes flex with fewer salt bridges, swaying easily at low kinetic energy. Membranes drip polyunsaturated tails, remaining fluid when water thickens. Desiccation tests all seals. Anhydrobionts load trehalose until cytoplasm vitrifies. Glass-like, preserving proteins and membranes. LEA proteins coat macromolecules, preventing aggregation. When water returns, glass melts. Metabolism lights as if the pause was a breath. Radiation cuts deep. Deinococcus radiodurans answers with rings of chromosome copies. Fragments find twins by overlapping ends. DNA repair, enzymes stitch mosaics into wholeness. Manganese peptide complexes quench radical oxygen, protecting proteins so repair can proceed. Pressure in trenches squeezes enzymes. Piezophiles insert small cavities in protein cores, giving space to compress. Membranes enrich unsaturated lipids, staying liquid under tons of water. Respiratory chains swap pressure-stable cytochromes. Each stress leaves chemical fingerprints inside the cell. Phosphorylation cascades. PP-GPP spikes. CAMP rises when glucose falls. C-DGMP climbs as surfaces are sensed. CDAMP whispers osmotic strain. Second messengers relay outside whispers into genetic reply. The orchestra never shouts. It murmurs. A gene turns on, another dims. Chaperones yawn awake, ribosomes hush, pumps reverse. Thousands of changes, each minor. Some to resilience. Imagine a desert grain glowing with afternoon heat. Inside, a single bacterium folds chaperonins around a drying enzyme. Night will cool the sand, dew may seep. The cell will wake and divide. Picture a vent plume surging with sulfide. Archaeal membranes hold. Reverse gyrase tightens DNA. Self-reduction enzymes breathe out energy that warms the tiny interior, just as brine tries to cook it. Envision a leaf after rain. Water beads roll, carrying nutrients. Pseudomonas cells taste iron scarcity. Siderophores bloom like invisible perfume. Scavenging rust flakes for growth. Sunlight dries the surface. PP-GPP rises. Cells slow but stay ready. Every stress response is rehearsal for change. Microbes accept impermanence. They bend, never break, flowing through centuries of flux. While you rest, stresses shift in your own body. Oxygen spikes near lung tissue, dips in your colon. And pH slides after meals. Antibiotics from forgotten doses linger. Your gut microbes adapt in silence, balancing protection and persistence. So you wake whole. The planet itself is stress. Day and night. Season and storm. Volcano and glacier. Microbes met them all long before animals dreamed. Their strategies became the toolkit of life. Thus, in every corner, glacier, gut, geyser, grave, stress pulls a chemical string. The microbe feels the tug. It answers with chaperone, pump, switch, splice, pause, or bloom. No complaint, only quiet resilience. Close your eyes. Let the thought drift. Billions of unseen cells, adjusting, repairing, enduring. Their calm replies to stress echo as a lullaby, reminding you that adaptation is gentle, patient, and always near. Sleep knowing the smallest lives. Carry on through every trial, holding the fabric of Earth together, one silent response at a time. Night settles over the microscopic world, yet no dusk or dawn touches the cells themselves. Their realm is lit instead by chemistry, by gradients, by pulses of ions slipping through membranes like moonlight on water. A single microbe drifts in a silent drop. To you it is smaller than a whisper. To itself it is an ocean of inside and outside, a cosmos of surfaces and depths. Membrane phospholipids sway, soft tails brushing one another in liquid dance. Embedded channels open, close, open again, letting a proton glide home, letting a sugar step inside, letting a sodium slip away. Ribosomes click softly, stringing amino acids into coils and sheets. Enzymes embrace substrate in perfect hush. Reshape them, release them, wait for another. DNA rests in gentle loops. Polymerase strolls along the strand, copying letters older than mountains. Behind, repair enzymes trail like careful caretakers, checking, straightening, mending the helix's quiet script. A faint shortage of carbon passes through the drop. Sensors feel the change. Alarmants rise only enough to slow growth. No panic, only measured breath. Minutes later, a trace of phosphate drifts near. Transporters sample, capture, share the gain across the tiny cytoplasm. Far away, another cell divides. Its wall constricts with patient precision. Two daughters emerge, each half the size, each already alive in full. Somewhere deeper, a spore still waits. Decades of dryness have not dimmed the genome inside. Moisture will kiss the coat one day. The inner membrane will soften. The scroll will unroll. Life will recommence from pores, within a biofilm's mattress of slime. Crowds of cells trade whispers of autoinducer. When the quorum feels right, genes for dispersal rise like a tide. Edge cells loosen, peel away, float off to find fresh stone or leaf. In the open water, a cyanobacterium greets dawn. Pigments collect photons, blue, red, green, passing energy downhill to centers that split water, lift electrons, write sugar from air. Oxygen sighs into the liquid, rises as silent bubbles, feeds unseen neighbors who breathe it in darkness. Between grains of beach sand, an archaeon reduces sulfur, exhaling sulfide that feeds still others. Cycles twine, carbon to nitrogen to sulfur to iron, threads braided by countless cells that will never meet, yet sustain one another without knowing. Every fraction of space is filled with this weaving. On your fingertip, in your breath, inside the creases of a blanket, life's smallest forms persist. Quiet, numerous, enduring. They do not seek applause. They do not craft monuments. Their triumph is to continue, to adjust, to share the same slow beat that began before continents parted. Listen for a moment to that beat, not with ears, but with calm imagination. Feel it as a distant surf of molecular motion, a tide that never rests, needing no light to flow. Breathe in. Countless cells enter, each carrying its own harmony of metabolism. Breathe out. Others depart, their work for you complete. When storms scour mountains, microbes in cracks cling on. When glaciers grind valleys, microbes beneath ice sip ancient minerals. When deserts bake under noon sun, cells hide in shadows of grains. Waiting, always waiting, for the next cool night's dew. They teach patience without words. They teach resilience without effort. They teach that life's grandeur can be measured in nanometers and zeptoliters, as surely as in redwoods or galaxies. Let that lesson settle over you now. Imagine the planet as a blue marble, encased in a faint, living haze of microbes. In ocean, air, soil, stone, a veil so thin you cannot see it, yet so vital that without it, nothing larger could stand. Feel your own heart slow a little. Feel your breath deepen. Know that the same ATP turning in their cytoplasm turns in your cells too. The same genetic letters, the same elemental atoms, passed along through eons. From microbe to fern to fish to human, rest quietly within you tonight. If your eyelids grow heavy, let them close. The story will go on without observation. Enzymes will keep folding. Protons will keep falling. DNA will keep humming its voiceless tune. And while you drift into gentle sleep, millions of unseen companions will continue their unending labor, stitching the world together, one tiny, tireless step at a time. Rest knowing the smallest things. Have everything in hand,